So we were dealing with the urea cycle in the previous slide, but I did not tell you anything about the enzymes involved in urea cycle. Instead, we just labeled these stages as 1, 2, 3, 4 like that, which are the enzymes catalyzing each of the stages. See, the first reaction is catalyzed by ornithine transcarbamylase. Which is that reaction? First reaction is actually the reaction between ornithine and carbamyl phosphate to form citrulline. Where is it taking place? Inside the mitochondria. That reaction is catalyzed by ornithine transcarbamylase. And what about the second reaction? It is catalyzed by argininosuccinate dehydrogenase. Catalyzed by argininosuccinate dehydrogenase. Argininosuccinate synthetase. Argininosuccinate synthetase. Where is the reaction taking place? Inside the cytoplasm. And you know that argininosuccinate is made. The third reaction is catalyzed by argininosuccinase. What is that reaction? Third reaction. Argininosuccinate is broken down into fumarate and arginine, taking place inside the cytoplasm. This reaction is catalyzed by which enzyme? Argininosuccinase enzyme. And the fourth enzyme, I already told you, arginase. It is the enzyme that acts upon arginine. Arginase acts upon arginine, releasing urea, and the other product is ornithine. This ornithine is transported into the mitochondrial matrix, and the reaction is actually taking place like that. Okay, now we will come back. This is the summary of the amino acid catabolism. Here you can see some of the amino acids are given in a blue colored box because those amino acids are ketogenic in nature, which means by their breakdown ketone bodies, for example acetyl coenzyme A or acetoacetyl coenzyme A, etc., are produced, and these ketone bodies in turn are used in a variety of chemical reactions, while the remaining amino acids are given in such a colored box. What do we call that color? Is it a red color? Okay, they are given in a different color. Okay, red color. And why? It's because they are glucogenic in nature, which means they break down into some intermediates which are going to be converted into glucose by means of gluconeogenesis. You see here alanine, cysteine, glycine, etc. are converted into pyruvate. You know that the pyruvate can be converted into glucose by means of gluconeogenesis. Asparagine and aspartate are converted into oxaloacetate, which can be converted into glucose by gluconeogenesis. So the amino acids present in these red boxes are converted into glucose, so they are glucogenic in nature, while the amino acids given in these blue boxes are converted into ketone bodies, so they are ketogenic in nature. But there is a problem. Some amino acids are listed more than once. For example, you see here leucine, lysine. Here also you can see leucine. Okay, these amino acids are shown more than once. You know why? Because different parts of their carbon skeleton are degraded to different end products. Different parts of their carbon skeleton are degraded to different end products. That's the reason. And one more feature is there. Leucine and lysine, they are exclusively ketogenic in nature. Leucine and lysine are exclusively ketogenic in nature. Otherwise, you look here, tryptophan. It is shown here to be glucogenic in nature. But you see here, tryptophan is there in this blue box also, which means it is shown as ketogenic. So tryptophan is both glucogenic and ketogenic. Similarly, what about 309 you see here? It is seen here also. So some amino acids are both glucogenic and ketogenic. And there is a topic in your syllabus, Cori cycle. It's very simple. When you are actively engaged in exercise, muscular exercise, you know that glycogen is broken down into glucose, glucose is broken down into pyruvate, pyruvate is further broken down into lactate, and you are respiring anaerobically. In anaerobic respiration, for example, you are running very fast and your muscles are aspiring anaerobically, lactate is produced. This lactate is transported through the blood into the liver. Lactate is transported through the blood from the muscle to the liver. In the liver, ATP is utilized and it is transformed back into glucose. By utilizing ATP, lactate is transformed back into glucose. And the glucose is transported by means of the blood supply into the muscle. Glucose is transported from the liver into the muscle and it is supplied for exercise. So this type of reaction, this pathway is actually called Cori cycle. This is essential for the survival of the organism. And some of the amino acids are classified as non-essential, while some others are classified as essential. You know why? Actually, both are required for the normal life. Some are classified as essential because we don't have the enzymes to prepare these amino acids from the others. So we need to get these amino acids from the diet. A dietary shortage of these amino acids will lead to deficiency symptoms. So these are the essential amino acids and we should get them through our diet. While these amino acids are called non-essential because we have the enzymes to prepare them in our body. So even if we are not getting through the diet, even if we are not getting those amino acids through the diet, nothing will happen to us because our body can synthesize it from the raw materials using the particular enzymes present. Why some others are conditionally essential? It's very simple. Required to some degree in the young and growing stage and during some diseases also. Otherwise, we can make them in our body. So this is how we classify enzymes into, we classify amino acids into non-essential and essential. That is the end of the chapter.